What was the availability of the biological material, and what were the difficulties to get or collect the material?
Xipholis ampelinus is a pathogen of vines. In the UK, there was no vegetative material present over the period of the TPS, so we had to use vine stem material. As you can imagine, this is a particularly tough material, so in order to produce a homogeneous sample, we had to try a number of homogenization methods to be able to grind the stem up enough to produce large quantities of lysate. The method we found most effective was to cut the stem material up using secateurs and place these pieces, small pieces, into large Nalgene bottles containing large ball bearings with a small amount of buffer. We then placed these samples on a paint shaker machine and ground them for just over 10 minutes, adding additional buffer halfway through the grinding process. Samples were later spun and a clear lysate produced and pulled into a single batch ready for spiking with Xipholis ampelinus cells.
So we were lucky because a lot of isolates for tosporviruses are in DSMZ collection. This is German collection. In addition to a good collaboration with many laboratories around the world, we were able to collect many different isolates.
For Plumppox virus, we maintain a couple of isolates in our own collection. But furthermore, we ask different laboratories and institutions to share their isolates, and we received isolates from collections from Hungary and Italy and France, Russia, and the Netherlands. So we didn't really encounter any problems in receiving extra isolates, and it went pretty well, actually.
After the first outbreaks in Italy, infected material was provided to our laboratory for confirmatory tests. This allowed us to have different local isolate as leaves, fruit and seeds, which were maintained dehydrated or frozen. All those isolates were artificially inoculated on tomato and pepper plants to increase the amount of biological material available for pretest and TPS. The amount of the obtained infected plants was sufficient for preparing all the reference material to be distributed among the participants. Other tobamovirus used for testing specificity were bought at DSMZ.
We have a collection of Bursiphalancus that we use as controls in routine analysis of our validation test. Bursiphalancus grow very well on Botryoptenia in Petri dish, so it wasn't difficult to find material for this TPS.
It wasn't possible to source naturally infected material for the test performance study, so we ended up just using pure cultures and extracted DNA, and we also decided not to include, not to spike material because of difficulties in the type of host material. So for this test performance study, it was purely pure cultures and DNA that was dispatched and no host material, just because of the difficulties in the matrix and being able to source naturally infected material.
What was the composition of the panel of samples?
Composition of the panel of samples was designed in such a way that it improved our knowledge of the specific tests and host plants included. So for example, for Erwinia amylovera, we know that there's a lot of data on apples, but we also wanted to generate new data on pears, on certain ornamental plants, and we've also included those hosts in the test panel. While for Pantoja stevarti, we know that the challenge is to be able to detect low levels of the pathogens and to differentiate between the two subspecies that can occur in such samples, subspecies stevarti and subspecies endogenous. So in that case, we've had samples spiked with low levels of the target, and also some samples containing subspecies endogenous, which is cross-reacting in many of the molecular tests for detection.
The panel was composed of 11 samples in duplicate for a total of 22 samples. We included 5 negative samples in duplicate, 5 positive samples in duplicate, each belonging to a different isolate and strain of the target, and 2 positive samples in duplicate corresponding to 2 deletion levels of one of the previous isolates.
The composition of the panel of samples was, we had in total 16 samples. We had a panel of 8 DNA extracts and a panel of 8 spiked wood material.
The panel of sample was prepared in accordance to the guideline of the Work Package 2 teams and TPS Round 1 organizer. Specifically, 9 samples were included in the panel, some in double and some in triplicates. The panel included leaves and fruits of tomato and pepper plants and was composed of 2 healthy samples, 1 infected samples at high level of dilution, 1 at low, 1 at medium concentration. For a total of 22 blind test items. We also provided the isolation and amplification controls.
Which samples were used to evaluate which criteria?
So with both test performance studies, we were mostly focusing on analytical sensitivity, analytical and diagnostic sensitivity of the tests, but we had some specific samples, as I mentioned, for example, for Pantoja stevarti, samples that did cross-react and could give us information on both on analytical specificity and diagnostic specificity of the different tests. In all TPS studies, we have also included controls at different levels. So in all our cases for test performance studies on molecular tests, participants also did DNA extraction, and we have provided controls for both DNA extractions and for molecular tests themselves. And these were basically the results that we used to judge whether the data sets the participants submitted were fine or not. If the controls did not have the correct results, the data sets were excluded.
So the main criteria we determined were the concordance or accuracy. So this means is to what extent were the results in agreement with the expected outcome that we determined in our preliminary studies. And furthermore, we looked at diagnostic sensitivity and specificity. And the first one, the diagnostic sensitivity, refers to the test ability to detect the target. And in case of sensitivity, we are talking the ability to not detect the non-target. So those were the 3 main criteria we looked at.
So we use target samples to evaluate diagnostic sensitivity and non-target samples to evaluate the diagnostic specificity. And the positive samples at different level of concentration were used to evaluate the probability of detection, and the accordance, also called repeatability for qualitative tests, and concordance, also called reproducibility for qualitative tests.
Did you encounter any logistic or regulatory problem?
Yes, so the regulatory status of Xiphilis ampellinus changed sort of prior to the TPS. In England, we no longer required a letter of authority to receive Xiphilis ampellinus samples. However, a number of the participant countries still did when they checked with their plant health regulator. Also, obviously, the lockdown had a big impact on the completion of the testing within the time frame and resulted in one of the participants dropping out, unfortunately.
Yes, our panel of samples included also one quarantine virus. This was Chrysanthemum stem necrosis virus. Therefore, letter of authority was required. I must say that some labs had big problems with getting the letter of authority in time. And I think that one lab was even not able to get it, most probably due to COVID epidemics.
To ship all the samples, a letter of authority was required. Unfortunately, not all the selected participants were able to have it in time. The emergence COVID-19 exacerbated this problem. We sent everything in dry ice, but it posed some logistic problem to send the material to New Zealand, and we circumvent this problem by shipping samples at room temperature. At the end of the TPS, all the participants are we are able to send materials and reagents to all the participants.
Some laboratories struggled to get their letters of authority in time for the planned dispatch date, so some samples were dispatched a little bit later than was originally planned, and I think one laboratory we just dispatched DNA to them because they couldn't get their letter of authority approved.